How's it going, everybody? My name is Dr. Dean Fido, and I'm the program lead for our forensic psychology program at the University of Derby. And I'm a former student and PhD student of Nottingham Trent University. Thank you very much for inviting me for this talk at the APE conference, and I hope you're all having an excellent conference so far. Throughout this session, I'm going to be talking to you about one of our papers which we've recently had published in Personality and Individual Differences with my wonderful colleagues at Nottingham Trent University, labelled Don't Slap the Fish. The full title of which is on the right hand side. So throughout this session, we will establish why we care about omega-3 fatty acids in forensic psychology. We're going to present our methodology and also our findings and future research over this two-study paper. Now, of course, I'm not going to be at the session today to help answer any of your questions. My Twitter handle is on the left-hand side and also my email at the end of the session should you have any questions. I also think that Alex Sumich will also be around to answer some of your questions that you might have about this paper. Okay, so this paper was born out of the first paper that I ever had published, which was entitled The Electrophysiological Study of the Violence Inhibition Mechanism in Relation to Callous Emotional and Aggressive Traits. And the study itself centered around the violence inhibition mechanism, which, if we boil that down, essentially what this means is it's an innate developmental mechanism within the human body, whereby if you're behaving in an antisocial and aggressive manner, if you're then faced with distress stimuli, such as fearful faces or fearful voices or sad faces and voices, this will help to invoke empathic loops, which will ultimately, in typically developing individuals, lead us to freezing or inhibiting that behavior, learning not to carry out that behavior in the future. However, what we see in individuals with higher levels of aggression or psychopathic personality traits is that this innate mechanism is atypical, and as such, they oftentimes carry on this aggressive behavior even when faced with these distressful cues. The paradigm that we used in this initial study was an adaptation of a stop-go paradigm, whereby individuals were asked to respond to aggressive stimuli in the form of a button press, and then subsequently were asked to stop this behavior before they actually pressed the button if they were subsequently faced with a fearful or sad facial stimuli. If the subsequent facial stimuli was anything other than a fearful or sad distress cue, they were just asked to carry on this button press. In terms of wider methodology, we used EEG measures from the scalp in order to identify electrical indices of both face processing and motor extinction to see whether aggressive or callous unemotional traits impacted these distinct aspects of the violence inhibition mechanism. For those of you watching this video on YouTube, I've linked the paper down below, or for those of you watching at the conference, if you'd like a copy of that paper, please do get in touch. So moving on to our interest in omega-3 fatty acids. Now, throughout this presentation, I'm going to be talking about omega-3 as a whole, just to make sure it's accessible to a wider audience, but the literature that we describe throughout focuses on a special strand of omega-3, EPA, which is also the type of omega-3 which we measure specifically within our research. Broadly, deficits in omega-3 rich diet has been implicated in violence and maladaptive behavior. Moreover, it has been inversely associated with self-reported aggression, anger, and also callous unemotional traits, specifically this latter part in children with ADHD. Of interest, supplementation of omega-3, again EPA, has been associated with reductions in physical aggression, anger, and rule-breaking behavior in forensic settings, meaning that there is definitely scope for the implementation of omega-3 rich diets in attenuating this deviant and maladaptive behavior. However, in terms of its associations with ERPs, very little research explored the utility of omega-3s. As mentioned before, and in reference to the violence inhibition mechanism, we can split the processes into two. First, face processing, which we know is inversely associated with some, but not all, psychopathic related traits. However, interestingly, blood levels of omega-3 inversely associated with M170 amplitudes to sad faces. On a practical level, this could indicate that the richer one's diet is in omega-3, specifically EPA, the fewer cognitive processes are required to process sad and distressful faces. When exploring electrophysiological responses to stopping behavior and ceasing and inhibiting aggressive acts, we know from our own research that the P300, this later cognitive potential, is inversely associated with one's externalizing disorders and self-reported aggressive behavior. Of interest again, we see greater cognitive resources available to ones inhibiting their behavior following omega-3 supplementation. However, when we boil down this information, we see a distinct lack of research triangulating these findings within a healthy sample. As such, we explored this within our 2-study paper. In the first, we looked for associations between omega-3 self-reported intake and aggression and callous and emotional traits. Although our sample was quite limited in terms of both number and distribution in terms of sex, what we did identify was that the higher levels of self-reported omega-3 intake in our population, the lower self-reported levels of physical aggression. We opted for measures of physical aggression over other types of aggression, as this better indexed the violence inhibition mechanism. There were no significant relationships between omega-3 intake and callous unemotional traits. As a whole, and another caveat to our study, omega-3 intake was relatively low throughout both of our independent samples. In study 2, we added to this using EEG data, as well as the FAST task that we drew from our previous study. After co-varying out alcohol and substance misuse, both factors known to attenuate omega-3 diets, we found that individuals reporting greater omega-3 intake had higher stop P300 responses to both fearful and sad distress cues. Moreover, on a behavioural level, we could see that these cognitive resources actually back-ended onto quicker stopping times and greater stopping success. Our take-home findings from this research were that intake of omega-3, specifically EPA, was consistently correlated with reduced physical aggression, and this association was likely mediated by a positive association between omega-3 intake and stop P300 amplitude and proficiency. Of interest, although there was a little research supporting the potential association between omega-3 and callous and emotional and psychopathic traits, omega-3 intake was neither associated with callous and emotional traits nor N170 responses to facial affect. Now, there are quite a lot of limitations within this study, and here are the top 3 for me. First of all, there was no blood quantifications of omega-3 intake. However, previous research has shown very high correlations between self-reported omega-3 intake and blood levels. Moreover, we know that omega-3 can be impacted by aspects which were unmeasured in our study. So, for example, micronutrients. And finally, if we are trying to make associations between omega-3 intake and behaviors mediated by cognitive processing and brain structure, then a longer term or historical measure of omega-3 could be helpful to specify exact neural changes and when they were mapped and impacted. Nevertheless, we would support future research in assessing the role of omega-3 supplementation in bringing about change in behavior and aggression. And that very much brings us to the end of this whistle-stop tour. Finally, I'd like to thank my collaborators on the project, as well as our industry partner, Terje Meyerwald, and also the funding that we received for the project from Nutrimed and my Vice Chancellor's bursary at NTU. So before I say goodbye, I've left my contact details at the bottom of the slide, as well as my Twitter handle on the left-hand side, should you have any questions or comments. But for now, if you're at the APE conference, I'll leave you in the hands of Alex Sumich to answer any questions you might have. Have a great rest of your day.